Welcome everyone to our meeting on GenZ of FSHD, FSHD 101. We have our two presenters with us today, Brittain and Justin Cohen. Brittain grew up in Tampa, Florida, and graduated from the University of South Florida with an undergraduate degree in biochemistry and in cell and molecular biology. He completed a master's degree in medical microbiology from the University of Florida before beginning his PhD work at the University of Nevada, Reno. He's currently a graduate research assistant in the lab of Drs. Peter and Takako Jones. Justin Cohen is a postdoctoral associate in the LEC lab. He completed his BS at Tufts University and his PhD in the molecular cell biology and genetics program at Drexel University. Now in the LEC laboratory at Yale School of Medicine, Justin studies FSHD, a topic close to his heart as he's also a patient. Justin is hopeful that his work in the lab will contribute towards a therapy for all FSHD patients. So I'll leave the floor to them two to now present.
Thank you, Eloise. I'll be presenting first. My topic is on clinical presentations of FSHD. Let me start my presentation and share my screen with you all. All right, Eloise, thank you for that kind introduction. My name is Brittain again, and I am also a patient. I was diagnosed at 10 years old and have been involved with the FSH Society since shortly after that. My talk today is going to go back to the beginning with the history of FSHD. Two defining physicians, two French physicians, Louis Landouzy and Joseph Jules Jagerin, did clinical work on FSHD starting in 1884. They first coined the Landouzy-Jagerin myopathy as part of their case studies. Their case studies involved work with a family and more particularly a young man that had FSHD or potential FSHD at the time. In their research, which this is a case study presented here, they describe it as a hereditary myopathy with influence on the face without influence on the nervous system. They go on to talk about the characteristic facial involvement and upper arm involvement that we're all familiar with FSHD. So what makes a clinical presentation of FSHD? First, it's the muscle weakness. There's a large amount of variability in the muscle effects of this condition, but primarily the earlier involvements, as seen in this diagram, are seen in the face, the tibialis anterior or lower leg muscles, and then the muscles of the shoulder and upper arm muscles. So this will change over time, and again, the variability on this condition is high, but there's characteristic on the foot drop and the overall muscle weaknesses specific in the right side on many people with this condition. So non-muscle features. This facioscapulohumeral muscular dystrophy also presents with bone and vasculature. These are low penetrant issues, which means they're not displayed in all patients, but they can affect the, as you see in the upper left side, the bones of the spine, also the bones of the sternum and rib cage in a condition called pectus excavatum. There's also a high frequency hearing loss seen in a fair amount of patients. So that would be normal hearing is between 20 and 20,000 hertz. And the high range of that is the 2,000 to 8,000 plus hertz. Not all patients, but some patients will show decreased hearing within that range. Respiratory insufficiency is there's a muscular component, but there's also a non-muscular component that is shown in FSH. The patients there will have decreased just breathing capacity, and also there's retinal vascular disease. It's a tortuosity or retinal vasculopathy seen in the eyes in some patients. So those are the non-muscle features that are seen in some patients, in addition to the overall muscular weakness. Prevalence of FSHD. So this is something that's been further refined over time, especially with the enlightenment of genetic diagnostics and the overall ability to find patient population in several different global kind of fields. So this was based on a case study out of the Netherlands. They found that roughly 1 in 8,000 individuals are affected by FSH, which puts it up there with myotonic and Duchenne muscular dystrophy, which myotonic's roughly 8,000 individuals, and Duchenne's is anywhere from 3,000 to 6,000. 1 in every 3,000 to 6,000 individuals affected. So you can see there's a lot of individuals that are affected by this disease. There's a lot of either misdiagnostics or there's patients that are unaware of their condition, and so this is likely a somewhat depressed statistic. And please feel free to stop me at any time if you have any questions. This is the clinical aspect, and Justin will carry on with the genetic aspects of the disease. On that topic, family history, I'll only be going over this briefly, as Justin will talk more of the genetics of the condition. But just that it's a autosomal dominant condition, which means it has an inherited component in roughly 2/3 of the cases. Using that, in clinic, they are able to track disease progression through families, which in my case, my mother was affected, and we suspect my grandmother was affected, and it made diagnostics or diagnosis a lot easier in this condition. So that is, there's a genetic component that 50% of the offspring of the individuals will have the affected allele, which is something Justin will mention. Diagnostics. So there's 4 modes of testing and identification of this condition. Neurologic testing, which focuses on muscle weakness and trends in gait and reflex response. There's genetic testing, which can be done primarily through Southern blot, which is a DNA analysis for the repeat basically repeat manifestation of this disease, also to be talked about by Justin. There's muscle biopsy, which it takes a punch of muscle component, and it can identify fat infiltration and just enlargement of muscle fibers, which are characteristic of this disease. And finally, blood testing, which involves serum creatine kinase, which is a muscle breakdown component that clinicians use to identify this condition. My diagnosis was brought about via Southern blot testing, which is the component of the genetic testing. It is done through several labs in the U.S. Orthotics and devices. So there's a range of assistive devices on the market. I am not a clinician myself, so I unfortunately cannot recognize or recommend any of these devices to you, but just so you know, they exist and they're out there for you. There's devices specifically to counter foot drop or head drop. There's also back braces, things that might help with overtaxed muscles to more effectively live daily life without that fatigue component. It was interesting. One of the first FSH meetings that I went to, I was hearing recommendations from other patients, and I heard about the one person's technique for they actually flipped around and would rest their feet on their headboard, and that would help with contractures that were caused by foot drop over time. So it's little kind of tips and little devices that we can use to help with ameliorating the conditions of this disease that are important. Another one that I found surprisingly helpful was a compression shirt I use since FSH is marked by scapular winging. I used compression shirts to kind of ameliorate the taxing effects of my scapula not being held to my rib cage as easily. So there are many of these. Another one is neck torsion machine, which is seen in the middle left, and that can be used to help with head drop and help ameliorate that condition. Community aspect. So I think it's an important mental health aspect of the clinic is being able to refer patients to the FSH community. That's primarily because there's a high degree of variability with this condition. So a lot of patients would benefit from discussions with other patients that have similar traits or similar difficulty to them. I think the global feature of this is brought about through the FSHD Champions, which kind of unites all the different nonprofits and organizations that do help with funding and research and patient outreach and advocacy for this condition, which is really an important part. The chapter network is more of a national ability to communicate between chapters and organize group events. And then the local chapter event, which mine is the Tampa chapter, or and more connected to the Southwest chapter in Florida. So I find it's definitely beneficial for me to have outreach to other patients in that community and be able to kind of personalize my treatment and my understanding of the disease through shared communication with other patients. Moving on, FSHD social media. So this is an aspect that I'm sure you all are much better at than I am, but it's important to have organizations like GenZ of FSHD, so you can not only communicate with one another, but organize groups and discuss through platforms much like this research innovations and just have a community to rely on when you need it. One of my favorites recently, I've been catching up on the FSHD Society Radio. It is a outreach program, and I know GenZ of FSHD has appeared in there, so I'm very happy to report that this is something that clinicians can understand and point patients, especially new patients, to when they're first joining the community. And finally, the Clinical Trial Research Network. This is a connected university and a hospital derived network. It's my alma mater, UF, is currently helping explore clinical trials therapies. Justin will discuss rather shortly, and all of these are important hubs for the disease research and places that offer increased resources to the patient population. So thank you all. That was my FSHD 101, at least the clinical aspects. We'll likely, unless anyone has any burning questions, save the questions for the end of Justin's presentation as well. So thank you very much.
Sure. All right. Thanks, everyone, for coming to the talk. So my name is Justin. I had FSHD. I was diagnosed when I was actually around 4 years old. I had to have a blood test sent to the Netherlands because at the time there wasn't any in the US. And so in my talk, I'm just going to go over the genetics and mechanism behind FSH. So, in order to do this, the first thing that we want to go over very quickly is the central dogma of biology, which is kind of the flow of information for how a gene goes from DNA to a protein. And the way that you can describe it is actually like a cookbook. So you can imagine if the DNA and the genome is like the cookbook itself, and like everything that you could possibly make. And then the gene itself, when it's expressed as RNA, can be the recipes for the specific product. This is basically the instruction that you need to do to make it. And then the final product itself would be the protein. So in this analogy, it'd be the cupcake from this recipe here. And there's lots of points of regulation along this pathway that's important to keep in mind when you think about how a gene is going to get expressed. And then the other thing to keep in mind is the DNA itself. So I'm sure most of you are familiar with the double helix cartoon of the DNA, but in reality, DNA doesn't really look like this. It's actually very, very tightly wound and compacted. So you do still have the double helix, but it tends to wind around a histone, the nucleosome protein that kind of beads on a string. So it just kind of circular lines around it, tighter and tighter. And then these beads also bond together, and they just lined up, becoming compacted and tighter and tighter and tighter and tighter until you lined up the chromosome. And in fact, the DNA wound up being so tightly compacted that if you were to take a single cell and flesh out your DNA from end to end, even though the cell is like not visible by eye. So yeah, it's very tight. And because of that, you need to realize that gene to become expressed, you need to open it up at the local level. So, can everyone still hear me all right? So now moving into the actual genetic of FSH. FSHD occurs along the tip of the chromosome, a region known as 4q35. And within this region, you have these repeats known as D4, D4. And in a healthy individual, you typically have anywhere from 11 to 100 of the repeats. And these repeats are very, very tightly packed. And on top of that, have these little residues, which called methylation, that act as a shield to even further minimize the gene expression that goes on in the region. So to FSHD to occur, this region needs to become open and compacted. And I think one way to actually visualize that I would like to try is the childhood blinky toy. So as you see, as you set the blinky toy in and out, it can sort of open up and you can sort of get into it. So you can sort of visualize like each link in the slinky as one of the repeats that we have. So looking at this in a little bit more detail, just as a reminder, we had the hundreds, 11 to 100 of these repeats in a healthy individual, but with FSHD1, this is about 95% of the cases, you usually have less than 11, somewhere between 1 to 10. So if you were to remove a bunch of these links and then put them in together while keeping the alternate phase in the thing, you could see that there's more depth in the sling due to the DNA basin, and that's kind of similar to what could be happening when you get through contraction. However, in 5% of the cases for FSHD, you don't have the repeat contraction, or at least not this many. Instead, you have a relatively healthy number of repeats, and instead, there is an external factor that allows the region to be opened up. So if you're wondering what the external factor is, remember in the beginning, I said that there was the methylation residues that act as a shield on the repeat. So in the case of FSHD2, the proteins, the enzymes that are responsible for the shield are mutated and wind up having recipe, and by having the decrease in methylation, you wind up opening up the region as well and allowing things inside it to be much more expressed. Dan mentioned about genetics, that if you had one of these mutations, you had about a 50% chance of inheriting it. If your parents had it, but it can occur de novo, as a random mutation as well. And that also happens in a lot of cases. FSHD, like in my case, for me, there's like no family history, so like they are random mutation. Okay, so now we're going to focus a little bit more specifically on the end part of these repeats. So if you just go to the very left of these repeats, you'll find out that it is truncated. And even at the end of this truncated region, you get this additional sequence that are known as a haplotype. And in a healthy individual, they call the sequence 4CD. And anyone that just has the 4CD sequence at the end does not have FSHD. So, as you said, but instead, there was a discovery that when they were looking at all the FSHD patients, they found that everybody that had FSHD, this is not a guarantee that they were symptomatic, but everyone that had FSHD had a 4QA sequence at the end instead of just 4QD. And the major discovery that occurred around a little more than 10 years ago was that the presence of this sequence at the end, combined with the opening up, the relaxation of the region, the disabilities, is what is the actual genetic cause of FSHD. So, looking at it in a little bit more detail, so why is this 4QA sequence important? So, to think about that, you need to remember the central diagram that you had, the DNA to RNA to protein. So, when a gene gets expressed to become from DNA to RNA, it becomes messenger RNA. And the messenger RNA has the tail at the very, very end, has a polyA tail that actually stabilizes the RNA. Without this tail, any RNA that is produced would be degraded and become unstable. So the 4QA sequence actually contains a code for the signal that is not present in those that have the 4QD. So what you can imagine is, if you go to the bacon, if you don't have this book here, you learn that it's just like this collapse and product that doesn't really work. In fact, it doesn't even make it to this step. So the thing to keep in mind is that the very last gene in that repeat that coincides with the tail codes to something called DAC4. And it is now known that this stable expression of DAC4 is the cause of FSHD due to the region being opened up enough to allow it to be expressed. So then what is DAC4 itself? So DAC4 is a protein that's normally only expressed during early embryonic development. So when a human embryo is around 4 cells, the DAC4 protein activates functions as a master switch, which turns on this network of genes required to produce all the cell types needed for development in the human body. So you can sort of see here that they activate a set of genes here that then go on to activate other genes. It'll basically cause this very wide signaling cascade. And this is because it's a transcription factor, which is a type of protein that will bind to the DNA in order to turn genes on and off, instead of other proteins that might function as a receptor or as a signaling molecule or something that like adds or takes away a residues in a protein. So in FSHD, all of these genes that DAC4 is activating, it's happening in childhood and beyond, like after you're born, when it's only supposed to be happening when you're an embryo. And this is very harmful for specialized cells, which can lead to the cells getting sick and dying. So just like the visualization here in this diagram, you have an embryonic cell here. Under normal conditions during development, it would you get this activation of embryonic genes, and it leads to differentiation into a specialized cell. But now, muscle cells activating all of these genes that are not supposed to be activated by DAC4 winds up being toxic, and it causes the muscle cells to die. So why now is DAC4 called FSHD? Because even though it can be toxic, DAC4 expression itself is very, very rare. Typically only around 1 in 1,000 nuclei in your muscles actually express it. So then why do we have such a devastating impact? One possibility is by looking at the structure of the muscle itself. So because FSHD primarily is affecting skeletal muscle, if you look at the skeletal muscle cells, so when they're on skeletal muscle, they can mature from this precursor. It tends to elongate and fuse with other neighboring muscle cells until it becomes the elongated muscle fiber that has a whole bunch of nuclei within it. So basically, you have one fiber that contains the equivalent of many different cells inside it. So even though DAC4 is only being expressed in like 1 in 1,000 nuclei, within the nuclei is still being contained within a fiber where all the genes that are expressed can spread to all of the other nuclei within the cell. And this toxicity can wind up killing the entire muscle fiber. And this is one possible reason why DAC4 can be so toxic to skeletal muscle even though we're not seeing as much of an effect in patients and on many of the other types of tissues. But this is just one theory. There's still a lot of research being done to figure out why this is the case. So I just want to summarize here. The FSHD is caused by this opening up of the region in the genome that's normally displayed in acid. And this can occur either through a reduction in the VT, FSHD1, or modification to loosen up the region, which is FSHD2. And when you pair this with the 4-TH design, this allows for stabilization of the dystrophy protein, which is the toxic protein for the disease. And while it's rarely expressed, it's possible that the morphology of the skeletal muscle fibers may be causal to FSHD. On my last slide, I just want to mention that now that we actually know the genetic basis of FSH, there is a lot of therapies currently in development that are targeting various aspects of the system of the dystrophy, including photons, which is probably heard about there's not an drug. And just keep in mind, I do have both listed here. The protein, but the mechanism is a little bit up in the air. It could be the protein, or it could be preventing dystrophy from being expressed in the first place earlier on. So there's a lot of biotech companies that are currently developing drugs to target after the therapeutics for FSHD. And there's even more being developed by research labs that are not quite at the pharmaceutical or biotech sector. So I just think this is very, very exciting that there's a lot of stuff to look forward to in the coming in the future, and that it's more than likely that one of these is going to be something that will be helpful. And yeah, just something I wanted to end on a positive note. Good. And with that, we can take any questions.
Thank you so much, Ben and Justin, for that. That was very, very interesting. It's funny because I've gone to a lot of these presentations and I always learn something new every time. So does anyone have any questions? I can start us off with a question. This is aligned kind of more with Justin's presentation, but either one of you can answer this. Does it only take one affected nucleus to kill the whole muscle cell, or like does more than one nucleus in one cell have to be affected for the whole cell to be destroyed?
I mean, we don't know what happens in patients, but like whenever we've done cell culture models where you have the myotube, and there is a model where you can sort of track ductal expression because it's tied to this fluorescent promoter, so you can see when it's being expressed. And we saw when you track that just the one nuclei being expressed was enough to kill the entire fiber.
Go ahead, Andrew.
I was wondering about like the efficacy of a drug called losmapimod that I've seen is I think having clinical trials right now. Like, has that been effective at all at inhibiting DUX4, or is that like not really something that will do anything?
Yeah, so they actually announced back in June the results of the clinical trial that they were doing for like losmapimod. So but before the clinical trial, when they were doing this in cell culture and animal models, they were showing that it was inhibiting DUX4, it was either preventing the expression and stopping the activity downstream of it. But when they did this in patients, they were not able to look directly at DUX4 expression. They were looking at the downstream targets, like some of the genes that DUX4 activate. These are known as biomarkers, and it's what they typically use as a proxy for DUX4 expression because DUX4 itself is usually not expressed very highly. But in that clinical trial, they were not able to see a difference in those biomarkers in the patient. And however, there are some caveats to that. The way that they extracted the muscle biopsy to do this is a needle biopsy. So because there's a lot of variation and variability and gene expression in FSH patients, even within the same patient, it's very possible that like when they're taking the tissue at baseline and then again at the endpoint of the clinical trial that it wasn't consistent. Like they could have taken a region that was either much higher or much lower in gene expression by chance. And so they are going to have to work that out a little bit. But there are some positive things that came out of the trial because even though we couldn't confirm an inhibition of the DUX4 expression itself, some patients that took the drug still showed a improvement in muscle function in some of the assays that they did. And when they did a MRI, they showed that the patients that were taking losmapimod had healthier muscles compared to the placebo. Like they looked at something called body infiltration. Basically, when the muscles are degenerating in FSH, it sort of starts turning into fat. So they saw that there was not as much of that in the patients that were taking losmapimod. So yeah, there was some promising effect with the drug, but it wasn't like a knockout process at all.
Right. Thank you.
Does anybody have any more questions? All right, well, I'll stop the recording.